PhD in physics at MIT, where he studies statistical mechanics and dynamics of surfaces and membranes. And then after a postdoc at Harvard's physics department, he eventually joined in 1995 UCSD's physics department. So if it's not clear, Terry is a card-carrying physicist, but today he's going to talk about some of his contributions to microbial systems biology. And so his lab has really led a lot of pioneering efforts into understanding bacterial growth phenomena that was first observed in the 1960s and 70s. And through this idea of proteome allocation, has really come up with laws for bacterial growth that every time I read his papers, I think that biology is perhaps as predictable as maybe mechanics or electricity magnetism. So Terry has won a number of major awards, just to name a few. He's been a fellow of the American Physical Society, fellow of the American Academy of Microbiology, and he's also a member of the National Academy of Sciences. So let's welcome Terry to UGA.
Thank Chris for the kind introduction and being a wonderful host. I'm very happy to be in Georgia. This is the first time in the state of Georgia, and learned from Chris that this place in Athens is the first public university in the country. It's great to be here. Okay, so let's get rid of and is there a way I can get rid of this thing? No, okay, never mind. Yeah. So as Chris saying, why is it not working? Okay, yeah. So I'm a physicist turned microbiologist, and actually, our lab, we call ourselves microbial bacterial physiology lab. Okay, and the physiology, we see it as sitting in between molecular biology on the one hand and ecology on the other. Whoa, it's not. Okay, so physiology, what do we study? We are interested in bacterial behavior, right? So exponentially growing cells, can talk about growth rate, can talk about growth yield, but there are many other phases of bacterial. Bacterial has many other states, and we're interested in all of these behaviors. And we're interested in this behavior in the context of the environment, right? And the nutrients, temperature, so forth. Now we would like to, so we also call ourselves quantitative biologists. Well, our goal is to have a quantitative understanding of, well, hopefully predictive understanding of the link from the green stuff to the blue stuff, environment to behavior. Okay. Well, that link is not so easy. The problem is that causally speaking, we know that the environment exerts their effects on molecules. And in principle, then you need to know how molecules respond to the environment, how the signals, how they interact, eventually lead to the behavior. Right? And that's a lot to ask, even for a simple organism like E. coli, not to say just generically. But then we have an existence proof that maybe all of this complicated molecular behavior can be simplified to allow us to have a predictive quantitative relation from environmental physiology. And an example is the monogrowth law, that the hyperbolic relationship of how growth rate depends on concentration. That's something he wrote about already in his thesis in the 1940s, right? So you might think about the monogrowth law just as a Michaelis relation for the uptake protein, and the uptake enzyme that depends, whose activity depends on the concentration outside. But it's far from that, because as bacteria grow at different rates, many things change. The protein composition, everything changes, including those uptake enzymes themselves. Okay, so it's very non-trivial to have a simple relation between this between nutrient and the growth rate. And nevertheless, there's a simple description. Right? So maybe there's a hope that such kind of relations, not the same form, but that we may have eventually one day understand also the effect of temperature, pH, and many of these ring variables, right? The effect on the growth rate. But that's just for exponential growth. So we're interested more than for other things of interest, of course, beyond exponential growth. For example, bacteria are not growing, and they are starved, stationary phase, and then how would the viability depend on these parameters, right? And they could be in stationary phase for different reasons, not starvation, but due to stress or whatever, and how does viability change? So these are all kind of the things that I'll take Monod's growth law as a example of the one day just like shooting for the moon if we have all of this understanding at some point. Of course, everything's a molecular biology underlies everything, but maybe there's also a simplified relation between the two. And ultimately, we ask why should there be any simple relations? Well, maybe philosophically, I would say maybe a simplified relation is also good for bacteria to navigate around. Okay, but we know at least in some cases we have simplified relations. Now these days, okay, until 7-8 years ago, the only organism we know how to spell is E. coli. Okay. And yeah, so I made my transition from theoretical physics to molecular biology, microbiology about 20 years ago. But for the first 10-15 years, we're working on just E. coli learning. Well, first 5 years we're just learning how to pipette. Okay. But the last 7-8 years, we were daring to go beyond E. coli, and so we very much like to move further to understand to do this sort of relating environment to not physiology but to ecology. But then I'm still at the very beginning of the growth curve of understanding some realistic ecosystem, right? And the system we are more working on is thanks to this Simon Foundation in collaboration with Marianne, together working on marine systems. Okay. But in the meantime, there's a lot to understand about the environment for a particular ecosystem. But in the meantime, what I would want to tell you today is that some other line of work is a different direction towards ecology that we're looking at how much of what we learn of the link between environment and physiology for exponential growing cells apply beyond E. coli to other types of bacteria. And so that's what I want to share with you. So let me start with what we know for E. coli. Okay, so there's this phenomena that we call microbial growth law that was discovered in the lab of Oli Moller in the late 50s, and then Neihard, Matsane was brought it to the other aspects of the E. coli in the 60s and 70s. So if you grow E. coli and Salmonella in different mediums, so the x-axis in each of these is the exponentially growing culture, and x-axis is the growth rate due to nutrients, different use of different nutrients. Y-axis is the ratio of the total RNA of the culture divided by the total protein of the culture, which back then in the 50s were some of the things people can already measure very well. Without at that time, yeah, they really don't know about genes and so forth. Like macroscopic measurement can be. So there's a linear relation. Okay, and that, yeah, if there's some kind of a hill curve, yeah, no, some complicated regulation. But if there's a linear relation, it's simple. It's not then so we're thinking about where it comes from. Well, Oli Moller also. What's going on? Sorry, I've got. Yeah, it's okay. Okay, so Oli Moller already had developed an understanding of this relation. And let me explain to you. First of all, the y-axis RNA protein ratio is a proxy for ribosome concentration. Okay, RNA, most of the stable RNA in E. coli, so ribosomal RNA, So it's a one-to-one relation from amount of ribosome, RNA to ribosome. And total protein is a proxy for cell volume, because first of all, the major component of biomass, bacterial biomass, protein, and then biomass density is the constant. Okay, so divide when you see everything divided by total protein is roughly divided by the cytoplasmic. So there's basically a conversion that we can tell. Okay, so RNA to protein ratio is proxy for ribosome concentration. So Muller's understanding of this linear relation is that, say, okay, assume all bacterial proteins are stable. It's pretty good approximation in these growth conditions. Over 95% of the protein is stable; they're not degraded. Okay, so protein accumulation is just a product of how many ribosomes there are and how fast they work. Okay, so then we can write down the equation: the exponential growth, then the total the rate of protein mass accumulation is this growth rate multiplied by the total amount of proteins in the cell. And then that should be given by number of ribosomes, each working at some rate epsilon. So that's how the epsilon is how fast each ribosome works. You can write the number of ribosomes as the total mass of ribosome divided by the molecular weight of a ribosome. Okay? And then you equate these two sides, you get a relation that the fraction of a total protein that's a ribosomal protein is proportional to growth rate, right? And with a proportionality constant that dependent on the elongation. And so that's Muller's explanation, rationalization of this linear relations. And in particular, right, so there's a expectation that the inverse slope of this line is proportional to the elongation with the ribosome. That is, so if ribosome elongates faster, then you should need fewer ribosomes to make the same amount of protein. So the line will be 5. Okay, so that's this picture. Now, when we first encountered this type of work, this was stuff done in the 60s. We were quite excited. Oh, that seems like something quite general. But then, you have a linear relation. There could be many ways, and it could just be a social kind of a rationalization. So we sought to look into a bit about this. In particular, we asked, well, if Muller is right, then if you change the speed of ribosome, this line should change, right? In a particular way. So postdoc mascot in the lab, that he's a mathematician, but to do a simple microbiology experiment. He did the same kind of experiment for a number of translational mutants, and it's indeed see that's still linear relation, but the slope change, right? And but then the ribosome speed of these mutants have been measured by people who first isolated these mutants. And the plot here is the inverse slope of these lines to the elongation speed. So these are in vitro elongation speed. It depends on how much substrate you put in. So the maximum one is when you have a saturating amount of tRNA in an amino acid. And there's the linear relation as expected. Moreover, if you work out the slope is just the molecular weight of the ribosome as predicted. Okay, so that convinced us that okay, Muller is really on the right track thinking about where this linear relation comes from. That is, the message is that ribosome's capacity is saturated. So if the cells have to grow twice as fast, okay, so that means in the same amount of time they may need to make twice as many proteins. You simply need to have twice as many ribosomes to make this. Okay, that's the upshot of this. Okay, and we'll see the amount of ribosome or translation-related proteins that's involved in the cell could be quite high, something as much as 40%. So then that means then when you change when a cell changes the growth rate between slow and fast, then the amount of protein that's need to be dedicated to ribosome is substantial, and that means other cellular components will be affected. Right? And that's very obvious when you look at the amount of the protein. So let me give you a specific example in particular. So now look beyond the ribosomes. Okay. So here we will show experiment where we change the different ways to change the medium to arrange for a range of growth rate. Okay, of a factor of 3 for change. What's shown in green are rich medium, just different grades of a rich medium. Various amino acid, nucleotides, so forth. And you see between the fastest minimal medium and kind of the fastest rich medium, there's a two-time change in the growth rate. Associated with that, there is a change of the ribosome amount. So in this case, it's not the RNA that's being shown here. What's plotted is so we do proteomics, quantitative proteomics, and so each protein you can get a measure of what fraction of that protein among the total protein. Okay, and you add up everything that's translation related. Okay? Most of it's ribosomal protein, but also elongation factor. So, and you see the fastest minimal medium and the rich medium implies a 13% change of the protein from 25% to 35%, something like that. Okay. All right. So that's 10-15% of the entire protein is a huge amount of protein. Okay. And so where does that come from? Well, one we can point to, so we have a proteomics, so we have a collection of thousands of proteins. And then what's plotted on this plot is a fractional protein that's devoted to biosynthesis. Okay. And so then we see that in the fastest minimal medium, where you need to make the most, the cell need to make all of the amino acid to fuel protein synthesis. It has to make lots of biosynthesis enzyme. And below that, there's a linear relation that's suggesting the process of the biosynthesis is that these reactions are enzyme limited, right? So to produce more, you have to make more enzymes. And but then in rich medium, where amino acids provided, you don't need to make these amino acids, so it's much reduced. That drop is equivalent to that. Okay, and I should make a side note here that you'll see a lot of plots of a mass fraction. The mass fraction is a convenient way to say, well, how much of a proteome is devoted to this, that. Okay. But it's more than that, because as I told you, total protein is a proxy for cell volume. So mass fraction is also a proxy for concentration, can convert 1-to-1 to concentration. Right? So you read this as concentration, and you really see that the flux of each amino acid synthesis pathway, which is proportional to the growth rate, right? Just related by the concentration. That's the interpretation for this linear relation in biosynthesis. Okay, but in rich medium, that's okay, so that's much change in biosynthesis. But if you look at the other section of the proteome, I could be using a pointer. You see that nucleotide synthesis is much less. What we're talking about is the 1-2%. These are absolute concentration, absolute scale. Okay, this is like 15% of the proteome. This is a few percent. Similarly, various energy, essential carbon monoatoms, and so forth. The changes all the time. Okay, so it's dominantly cells are shutting off amino acid biosynthesis and using these proteins to replace those spike ribosomes. Now, often microbiologists think, you ask, well, why enriched medium cell grow fast? Often people think, oh, it's the energy. Right? So rich medium provides more energy and so forth. But actually, if you do a careful accounting of the energy, see the energy is way in surplus. And so others have done an experiment where they delete the entire ATPase operon, which is which encodes the F0, F1 ATPase, that's the proton ATPase conversion system. Not only is the cell viable, seeing minimal medium, growth rate is not even affected. So in aerobic growth condition with plenty of carbon, energy is not an issue. It's the proteome that's limiting growth. Now coming back to this picture, we have a picture that in minimal medium. So this is a indication the proteome of the cell. We have a lot devoted to ribosomes. We have a lot devoted to biosynthesis, and then in rich medium, most of that biosynthesis is going to convert to translation. All right. So then you see that in principle, there's a problem if you, let's say, cells growing in rich medium and suddenly they transition to minimal medium, then they will need to make these biosynthetic enzymes in order to grow. But to make these enzymes, they need to have amino acids. But these enzymes are making amino acids. So there might be a problem, right? So you make a transition from one to the other, or cells may be stuck. They need to rely on the little bit of a basal level of these enzymes in each of the amino acid pathways, to regenerate these enzymes in order to grow. And we did a study of that. So keeping always the same carbon source, but then getting rid of amino acids at some point, time 0. Okay. And it doesn't matter which carbon source you use, basically there is for E. coli, there's a 2-hour lag from rich medium to minimal medium. Okay. And the various way to show that that this lag is just due to biosynthesis. Okay, in particular, if you supplement with, say, one of the amino acids, you see a shortening. Right? And we can also look at it from the recovery of the enzyme. So in this plot, so this is time after shift, you see how the enzyme level recover, and basically biosynthesis is the major one that has to recover. So the brown bar here is the new steady state. So basically, only after some time when these enzymes start recovering, the growth curve start picking up. Okay. Now, to look into this a bit more, we can pull down the enzyme, add together the enzyme devoted to each of the biosynthetic pathways, and you see that generally, of course, they all increase. But then if you look at the numbers, you see that some, like methionine pathway, from the rich medium, that's a blue line, the starting point, increase over 100-fold, whereas for the lysine pathway, it increases only 4 or 5-fold. Okay, so different amounts, different pathways, they have to recover by different amounts. And then if you pay attention to the time each pathway recovered, you see the ones that have to recover a lot, they are the ones that start pick up first, right? And the ones that's late, nothing happens to them until very late. Right? So this is a picture basically when the cell is switching to minimal medium, it has to use the little bit of a amino acid it can synthesize, okay, and it's putting to make the most needed enzyme first. In this case, it's a methionine, and the MET-E is the dominant enzyme in this pathway. This is the vitamin 12 independent pathway making methionine, very costly, and most of the resources first put into making this pathway, and when after this is no longer the bottleneck, then they start to work on the next one, and the next one you see orderly progression. The problem is typically you see orderly progression of which enzymes are made based on how much they're needed. Okay, all of these words I say can be turned into mathematics, and the red line is the prediction, not a prediction, it's actually a prediction of a model with no fitting parameters. The only input to this model, the model was actually a prediction because the model itself was actually introduced in paper 5 years before this paper. Okay, and then the only thing that the model needs to take is the initial and the final conditions. So we need to put in a steady state. Only thing it knows about the steady state concentrations. the first initial and the final, and also the initial and final growth rate and the linear relation of ribosome to no kinetic parameters needed. Right? This is a and you can do things in reverse, and it also makes the correct prediction in reverse. So this is an indication that none of this is a resource allocation based on how ribosomes are used. So all of this point is to say that the relation ribosome is very central, not only just for ribosome, but in terms of governing growth, and including growth transitions. So what I want to focus on today is, well, what happened to proteome allocation in other bacteria. Okay. So what I'm going to show you is a set of experiments where we look at a number of species. I'll focus on 3, but I'll show data for 6 species. Okay. Each with a number of medium, right? And then we will show you data for the different protein amount, right? And we'll do a comparison. So to the bioinformatics students, you're probably used to proteome comparison, right? I mean, that's very, but no, sorry, genome comparison. That's very. Okay, proteome comparisons are different because proteome depends on the growth condition. That's why we have to look at different growth conditions. You think about 2 kinds of comparison we'll make. One is maybe pick a special growth condition. In this case, let's say the rich medium. So for each species, for practical purpose, empirically speaking, there's some medium that people can get these species to grow very well on. Okay, we take these, and you can look at proteome comparison like a genome comparison. Okay, or you can take this pattern of expression like ribosome there. Okay, and then compare across species, and you need to do this for each different ribosome for biosynthesis, for energy synthesis, and so forth. Each can do a comparison. Right. So that's the 2 ways of doing comparison. And now there's one issue is that clearly growth rate has something to do with the protein, at least for E. coli. But growth rate of different species are very different. So how do we even make this comparison? There's a question. Okay, even for the same species at different temperature, growth rate, and of course different species grow at different preferred temperature. Right, so it's not trivial how even strategic how to compare the different proteins. All right, so we'll see how we take on these questions, and in the what I'll be talking about. Well, first we include E. coli, and then we're looking at the fast organism like liver nitrogens. When the record holder can grow as much as 2 times faster than E. coli, and we look at slower growing organisms. Not extremely slow, but in the fastest condition, there could be like 5 times slower than. Okay, so that makes up activity. All right. So, first question could be, well, is there still going to be a linear relation between ribosomes and the growth rate for this other organism? And I'll tell you, yes, we found linear relation, and no other people have found this kind of relation for other species before. Okay. But then maybe you think, well, that's it. Well, what else is there to talk about? Okay, let me illustrate for you the kind of there's some question remaining. Let's say the start here is the fastest medium, the fastest I can grow this organism in. Right, so it'll be 20 minutes of doubling for E. coli. I'll call this lambda star, the fastest growth rate, and the highest fraction of ribosome application, called the 5 star for ribosome. Okay. Now, if the linear relation holds, then the growth rate reachable should be given by 2 factors: how much ribosome and how fast each ribosome elongates. Right, the mass of ribosomes not that different from one species to another. All right, so then we have here significant nitrogen, which is growing much faster than E. coli. Okay, so how to get there? well, we have several options. Maybe vibrio nitrogen will have more ribosomes. Okay. Well, we got a problem because we already have close to 40% of the proteins in E. coli that are ribosome, right, translation related. So how much more can you push this? Right? Let's say we have 60%, 80% in vibrio nitrogens devoted to ribosome. Well, that's going to require very, very efficient the other enzymes doing other things that are needed for the cell wall synthesis and things like that, right? So if 80%, 60-80% devoted to ribosome, the other enzyme need to be much more efficient. We call that the cheetah sort of strategy, right? You become very lean elsewhere so that you can do very well. Alternatively, you can have they keep the same amount of ribosome, but you just have your enzyme faster. Theoretically, it's always possible, but how much faster can the enzyme be, right? So then that, in principle, if that's the case, then we're not comparing the cheetah and the human animal. We're comparing to different mechanisms of the locomotion, okay? But reality can lie somewhere in between. So then there's a question of a strategy, right? So if organisms have to move fast, or same as if they move, do they grow more slowly? What happens to these? Yeah, so what we find is surprising to me, okay? Both in where we find things changing and where things do not change. That was the title, surprising rigidity and the flexibility in the proteome allocation strategy. So let me start with something simple. E. coli and the Bacillus subtilis. We grew them both at 37 degrees and picked a number of rich medium, and they have similar growth rates. Okay, and if you take the RNA protein ratio, the Bacillus in green also form a linear relation. That's known. Okay. So the first curious question is, well, how does this green line compare to the E. coli? Okay. We know they're linear, both are linear, but they could be very different in practice. They're actually very close. Okay. So we know E. coli, the many experiments that establish this relation, E. coli linear relation, is basically due to ribosome. It's governed by the speed of ribosomes. Okay. And it's the same for Bacillus subtilis. Okay, never mind about this formula. If you support for the translational E. coli being measured in the previous work by us, so it actually is dependent on the growth medium. So very fast growing culture, fast growing cells, the ribosome speed is high, and if you limit the nutrient growth is slow, then there's a twofold change in growth rate. Okay, we understand that relation. It's a hyperbolic relation coming from ribosome. Never mind, it's not as so there's a relation. So then we measure the elongation rate for Bacillus subtilis. Okay, exactly overlapping. All right. So this is surprising? Well, maybe not. Right? Because in particular indicating that the maximum elongation rate in the rich condition where the substrate is saturated and everything, the same between E. coli and the Bacillus subtilis. Right? That's actually what we expect. Can argue that ribosome is a very important molecule for cells, for bacteria in particular, right? And presumably there's a lot of work, lots of thought in optimizing ribosome. Okay, so that may be what E. coli and Bacillus shares, a kind of a perfectly optimized ribosome that's as fast as it can go in people. All right, so with that in mind, let's look at the vibrio nitrogens. So vibrio nitrogens, as advertised, grows faster in a similar type of medium. It's in our hand they grow about 50% faster than E. coli and the bacillus at 37 degrees. So here I'm taking temperature out of it, or at 37. If you look at RNA protein ratio, they're actually similar. Okay, so the mentally you can expect if you plot RNA protein ratio versus elongation rate, they're going to be flatter. And this is indeed the case. Okay. So what that is suggesting is that okay, the same amount of ribosome, but then for liver nitrogens, it's 50% faster in growth. So that's suggesting that the elongation rate of ribosomes, VBACs, the ribosome is going 50% faster. So we directly measured it, and it's indeed 50% faster. No, for different each other. Okay, well, that raises all kinds of questions, right? I mean, these guys get good machinery, ribosome are fast. I mean, that's good, right? So why doesn't E. coli and bacillus also have that kind of a fast ribosome? And never mind about E. coli and bacillus, what about Vibrio cholera, which grows a similar rate as E. coli? No, other kinds of Vibrios, they don't have this kind of speed. All right. Mechanism, the whys aside, okay, we're looking at this behavior that was similar protein model, giving rise to different growth rate, different enzyme speed. It reminds us of something that's known for E. coli for a long time. So it was first reported by Fred Neihart that E. coli growing at different temperatures. Okay. They grow at different rate because of changing temperature, but the protein did not change. At that time, it was I think the first use of a 2D gel for proton, looking at proton. They don't know which protein, but they see the path is the same from between different temperatures. And if you do this so we repeated the experiments for E. coli at growing at different temperatures, and you see that indeed the slope are different, but the level of RNA protein is not that different. Okay, so for changing temperature, we understand the mechanism. Slower temperature, the kinetics are just slower due to the Arrhenius effect of the enzyme kinetics, and enzyme slower, and then if all the enzyme is slower, then the protein level does not change, and you just get slower. Okay. And let me just establish it a bit more. I mean, each of these linear relations, if we still take them to be given by this type of a relation to the elongation rate, then we would expect that the slope of these ribosome line to have this simple relation with the elongation rate, which we measured at different temperature, and indeed there's a linear relation. And also, since that the ribosome allocation itself does not change, then we expect that then the growth rate, since this factor doesn't change, we expect growth rate just to be proportional to the elongation rate. And indeed, this is the case for E. coli. We repeated this for Bacillus subtilis, exact same situation. Okay, so temperature dependence very, very straightforward. Now I sneaked in here Bacillus subtilis. You can actually Bacillus subtilis it can grow until 50 degrees. Okay, and at 43, 45 degrees, its growth rate is similar to Vibrio nitrogens. Yeah? And its elongation is similar to so that prompted us to think that maybe we can think about Vibrio nitrogens as effectively going at a higher temperature. Okay, it's at 37 degrees, but it looks like B subtilis is growing at a higher temperature. All right, so then we made the same kind of a plot that in LB, we take the maximum growth rate and maximum elongation rate and plotted them. The three red symbols are E. coli at different temperature, three green symbols are Bacillus subtilis at different temperature. That green point, that blue square is a Vibrio nitrogens. So here we see a relation that transcends phylogeny, right? Different organism, but make the same plot, they actually follow the same relation. Okay, and you don't need to worry about the name of the organism. Actually, they're just dictated by how fast the ribosomes. Okay, so then this is the case not only for the maximum rate. You can look at the slope of this ribosome line, and the product with the maximum elongation rate. See the linear relation. All right. So since we expect this to hold, right, then we say that if we plot all of these with growth rate scaled by the elongation rate, then all of these curves the points should collapse together on a single. And indeed, we see that's the case. So for all of these organisms at different temperature, the protein ratio versus the growth rate, when you scale by the elongation rate, all the same thing. Same happened to the elongation rate itself. Okay, when you plot it against everything normalized by how fast ribosome elongates, they all have the same relation. Different, right? Very different species. Right. So, this is these are evidence supporting that different organisms that have nothing to do with each other phylogenetically. Actually, there's something about them that the internal organization that make them behave this. All right. What about beyond ribosomes? So we did proteomics for these 3 species, and I'm going to show you a whole bunch of plots where the x-axis is the scaled growth rate. So we're already taking into account this now a trivial change just because the elongation rates are different. We normalize the growth rate by the elongation rate. What's shown here is just the translational machinery. So it's just that there's nothing new here because RNA is, I said before, RNA is a proxy for translation machinery. Here we're directly measuring the total amount of protein taken up by, well, actually also translation and transcription, but transcription is tiny amount, mostly translation. Okay, they're the same. Look at the biosynthesis, dominated by amino acid biosynthesis. Again, we see that. Okay, so the fill symbols here are rich medium, and open symbols are minimal medium. And minimal medium, we see again there's a linear relation between amount of the enzyme and the growth rate. And then there's a susceptibility request for different degree of rich medium. All right, so that's the case. And we have central carbon metabolism, TCA cycle glycolysis, and all that. Overall, there's not much of a growth rate dependence, but then it's not what the data is not just there's no growth rate dependence, but the values are certain. Okay, so what jumps out is that the values 15-20% here is dedicated to central carbon metabolism. Over here, in the fastest minimal medium, 20% of the proteome is dedicated to biosynthesis. These numbers do not change. Okay, and for ribosome, the fastest growth rate, 40% is dedicated to basically central dogma machinery. I said central carbon metabolism is constant, there's no growth rate dependence. But if you go into the subgroups, actually there's strong dependence. TCA enzymes increase slow growth, glycolysis increase the fast growth. They add up together to give you a very constant. And individually, these very different organisms devote the same amount of enzyme to these functions. You can even zoom in further to look at the individual pathway. Here's some example. Again, similarities. We have so here is a bird's eye view of a collection of a number of easily identifiable major pathways. And we see that there's a congruence of the allocation by different organisms. Ribosome is always the highest, and some low ones. So for example, what is this? Pentose pathway is maybe 30 times lower, and then some of this aspirin is even lower. Okay, so more or less the kind of they move together. There's certainly exceptions. So for example, tryptophan pathway, Bacillus subtilis was not expressed that, but that's because we're looking at the tryptophan autotroph, and we have to provide tryptophan in a medium for it to grow, and that's going to repress the tryptophan pathway, right? Other exceptions, motility is different, but then we're looking at the lab strains of E. coli and Bacillus, and lab strains typically we select for motility. That's for E. coli, we know how motility so selected. Then and a few other things like Bacillus subtilis, their biosynthesis is more highly repressed in LV. Now I told you before that in rich medium, the amount of a biosynthesis enzyme that's expressed is related to the recovery to minimal medium. Okay, and then we did those experiments to check, and we see that E. coli and the vibrio nitrogens, which have similar similarly, they have say about 50 minutes lag, whereas Bacillus subtilis, due to this very much more repressed biosynthesis, the lag is long. Okay, so all of these things. So the minor differences, but the major allocations are similar. All right, so to summarize, these are the sort of major results. I have some other stuff I want to talk about, but then just to recap what we've been talking about, right? So we do this proteome comparison, and the main results can be summarized by these two plots, right? We have a single time scale, basically that governs the differences. So growth rate change we account for by the single time scale, okay, change. Ribosome kinetics are different, and by implication, the biosynthesis enzyme kinetics are also different, but the same amount of enzyme produce different growth rates. So in minimal medium, they need to more fluxes. If vibrio nitrogen grows 50% faster, so the amino acid flux need to be 15% faster, but that's done by the same amount of enzyme, right? So it implies that kinetics is also faster. The kinetics of other enzyme other than the ribosome also fast. Very surprising is the rigidity of the amount of allocation. For some reason, they're just stuck on insisting on 40% maximum allocated to ribosome, 20% maximum allocated to biosynthesis. Didn't expect that. Don't know where that comes from, but it feels like we're looking at some kind of a master plan, and these guys are kind of following some master plan. Okay, and it's not necessarily related to phylogeny. Right. So we have many questions remaining. One is mechanistic, right? Whatever it is, operationally we know vibrio nitrogens are growing faster, it has a faster ribosome, and presumably faster biosynthetic enzyme as well. Where does that come from? It could be individual enzymes adapting. It could be another possibility is that things I suggested for you, the temperature change can also produce this type of thing. It could be that the vibrio nitrogen in its cytoplasm, it produces some kind of magic potion that makes it feel like it is a higher temperature. I don't know, right? So we're investigating these mechanisms, and the way we're approaching this is basically by swapping proteins and putting proteins of an enzyme that will from one medium into the cytoplasm of another. Look at the result, and it's still ongoing. Okay, then initial result is interesting. But then a larger question that we really don't know how to think of is evolution. So I'm a physicist coming to biology, and I read about what people write about like evolution, and often we see in the literature people saying that, oh, changing the amount of enzyme is easy, okay, but changing the property of enzymes is more difficult. Well, this comes largely from, say, developmental biology, where they're looking at the metazoan development and seeing cis-regulatory changes giving rise to different kinds. But also we have a laboratory scale evolution experiment on bacteria is maybe supporting similar type of picture. But what we see is completely the opposite, right? Kinetic is easy to change. but for what reason we really don't understand, amount is it's very stubbornly insisting. All right. Last few minutes, I want to talk about beyond these three organisms we're looking at. So students get excited. So, oh, maybe maybe it is true for large classical organism. I mean, we certainly pick ones not necessarily within simple phylogeny, but they behave the same way, right? So we start looking into other organisms, and I want to show you a particular one organism that's different, just to show that yes, certainly it's not like everything behaves this way. Okay, we picked. I want to talk about Pseudomonas. So we look at several Pseudomonas, but we have most data for this fluorescence, so I'll show data for that. It is Thermophilobacterium, rather closely related to the nitrogens E. coli. I should have shown this temperature response long ago. Okay, so so 37 degrees here, and the Pseudomonas often they grow at lower temperature, so 30 degrees for Pseudomonas fluorescens. And I should have said before that 37 degrees is not special, but not like in any particular weird range for living nitrogens or for okay, so they're in the operation of it's a good temperature to have. So now if you look at Pseudomonas, actually at 30 degrees, it's growing faster than E. coli. So we don't think about Pseudomonas as a superbug like living nitrogen growing very fast, but it's actually when you compare the same temperature, right? So I think that's the comparison to be made. It's a fast organism. All right. And if you look at its growth rate and elongation rate relation, they basically follow each other. Okay, nothing strange there. And if you look at the ribosome line, so that's the data I showed you for the three organisms previously, and Pseudomonas falls right along this line. So ribosome, it behaves the same way. Now, you look at the biosynthesis. Again, remind you that these are for the linear relation there is for minimal medium, and then you have rich medium where E. coli, circulars, and so forth are down-regulated with their biosynthesis. Pseudomonas doesn't believe in regulation. It doesn't need to regulate. Its enzyme is so fast that from rich medium to minimal medium, it can deal with the same amount of enzyme can support fast growth. And it's not just like so it gains like 10-15% of the protein just from that. And it's not just the biosynthesis, right? So here's a direct comparison of E. coli at 37 degrees and Pseudomonas fluorescens at 30. So there's already a disadvantage that E. coli gets to, at the high temperature, gets to grow faster. Nevertheless, right, Pseudomonas can deliver the same amino acid without adding up enzyme. Look at the transport. E. coli, when growth is limiting, it dials up the transport. That may have to do with its ecological niche that it's subscribing whatever, but Pseudomonas doesn't do that. ECA cycle, it doesn't regulate. Central carbon metabolism, it doesn't regulate. It's not just it doesn't regulate, it's keep at the minimum level. It doesn't need to have that much. So if you think about metabolism in this kind of a push-pull way, so enzyme is pulling metabolism, where substrate is pushing metabolism. So clearly they're operating very differently. Pseudomonas is working the push mechanism. So obviously more flux is needed at the higher growth rate in richer medium, but then Pseudomonas can get away with obviously this is suggesting Pseudomonas has much better enzyme that they don't need to upregulate it. They can just control by either allosteric or by substrate. All right, so I'm running out of time, so I'm just going to stop. I'm going to fast forward, and we actually look at other organisms, other So it turns out it's not, this is not unique to Pseudomonas, but we look at the number of acid preferring bacteria. Okay. There's Rhizobium, Meliloti, there's Mysobacterium magmatis. We also look at Marianne's favorite Rhodobacter pomeroy. They all behave this way, that they don't bother to regulate. And Rhodobacter is also acid preferring, right? And we actually started on this first from a Rhodobacter, right? Add vitamin B12, and it doesn't bother to down-regulate B12. But it's the same for all of these things, right? So we've seen that for many acid eaters, they have this behavior. Okay, so I'm gonna skip all that. Oh, and by the way, just to show that when we do the switch from rich to minimal medium, right? Which for the sugar eaters, there's this bad because they have to upregulate their synthesis. Well, acid eaters don't have to regulate their upregulate, and there's no bad. Just immediately switch. So, to summarize, we have these two classes of bacteria out of the 6 or 7 that we looked at. There's the sugar eaters that have this one way of allocating the proteome. We have these acid eaters have even more superior kinetics and doesn't bother to regulate many of the biosynthetic enzymes. They use their protein for other things that they don't have time to talk about. Okay, but they don't. They have a very fast biosynthesis. All right. So then finally, it's just the way I'm looking at this as a physicist looking at the microbial ecosystem. We're seeing, okay, so we're recognizing that these classes of organisms that have nothing to do with the first name, last name, but then they're behaving in kind of a type of a similar way, right? So we're suggesting there's an analogy to maybe a chemical system where you have different classes of chemical elements that's arranged not by the atomic number, but by the chemical properties. For example, the halides, alkalides, they could be very different by other factors, but then there's something similar about the chemistry. And here we suggest that sugar eaters and acid eaters also have this kind of a distinct but a very robust properties. All right, so this is all I have to say, and I want to acknowledge the people that did the work. So Melo Su and Shong Time, the 2 visiting students from China, actually really did all part of this work, especially measuring translation speed of different organisms. That's right. 80% of that work is just measuring the translation speed. Okay. And Chen Hao Wu was the physics student, graduate student that started this, and we did our proteomics with Rudy Wilson's lab. Thank you very much.
I have a few questions.
Sorry, longer than this.
Can you talk about really finding
the P is a ribosome model. So that's what ribosome model is what you exclude when you do transcriptomics. But that in this kind of assay, you basically want the total RNA is a possible. mRNA is only that finds.
I didn't catch you talking at all about the composition of the respiratory chain. For a while now, force the cell to use NPH1 instead of NPH2 that can affect the growth rate. Do you think that has any play on your cheetah versus normal state?
So these are we also do some investigation on that, and these have a smaller size. So we are looking at kind of from 10,000 miles away and kind of growth. So there's a big mystery in the energy budgets of it's a very well kept secret of people that study with energy budget doesn't work. If you count the ATP that's generated from the non-biosynthetic process, there's way too many ATP that's generated. Okay, and in fact, so you equalize using the inefficient energy waves, inefficient oscillating numbers.
From an energy perspective, from a rate perspective, like.
Yeah, no, but I'm just saying, if you compare the fluxes, there's way too many So we're going to get into with our picture of the why, but
I think to get Terry to his lunch, I think let's thank him and more questions after. Student lunch right after, so I think our students are.
Okay.
Especially Bill Baker. Yeah, that's great.
sorry. I was
That's right. You want to disconnect everything?
Yeah, I'll just I'll lab over here.